I'd now like to talk about why imatinib resistance occurs, and then also talk a little bit about some other diseases that are targeted by imatinib. So to summarize from my first part of the talk, I have noted that imatinib yields very high response rates with minimal toxicity in all phases of CML. In chronic phase patients, which comprise the majority of patients with CML, responses are quite durable and relapses only occur in somewhere between 10 to 15% of patients at 5 years. But in advanced phase, resistance is quite common and in fact a major problem. So just to remind you, these are relapses and disease progression in our chronic phase patients. So in our chronic phase patients, about 7% of patients have progressed to accelerated phase or blast crisis, and overall about 17% of patients, including those 7% of patients who progress to accelerated phase or blast crisis, have relapsed at 5 years. But it's a very different story in patients with more advanced disease. So at 4 years, if patients have failed interferon therapy, about a quarter have relapsed. And at 4 years, about close to 75 or 3/4 of patients with accelerated phase have relapsed. And in blast crisis, it's 95%. And in fact, the reality is in blast crisis, 80% will relapse within the first year and about 85 to 90% by the end of the second year. So clearly in the advanced phase of this disease, relapses are quite common. Now, as I mentioned, if we're going to understand what to do about relapses, much like understanding the cause of chronic myeloid leukemia, we have to understand the cause of relapse. So we have to start by asking the question, why do some patients relapse? So we've developed imatinib, a specific inhibitor of the Bcr-Abl kinase. So the first question I would ask is, in a patient who relapses, is the kinase still inhibited or not? Because if the kinase is inhibited and the patient relapses, it has to mean that now there's additional mutations besides Bcr-Abl that are driving the growth of the malignant cell. If, however, the kinase is no longer inhibited, it has to mean that somehow the drug is either pumped out of the cells by a drug efflux pump, maybe the target's been amplified or mutated, or perhaps the drug is metabolized, bound to proteins, or maybe some other mechanism. So how are we going to distinguish between these two? Well, we know that Bcr-Abl functions as a tyrosine kinase. And what does a tyrosine kinase do? It puts phosphate onto tyrosine residues of specific substrate proteins. As it turns out, the most heavily tyrosine phosphorylated protein in a CML patient is Bcr-Abl itself. But Bcr-Abl is an extremely unstable protein, and in fact, if we try to isolate it from a CML patient, we fail. But this other protein called Crackle turns out to be a much better marker. So what is Crackle? Crackle is an SH2-SH3 domain adapter protein. Actually, in 1994, our laboratory showed it was the most heavily tyrosine phosphorylated protein in CML patient samples. We then went on to show that it's a direct substrate of Bcr-Abl. In fact, one of the few times in which Crackle is tyrosine phosphorylated is in cells that express Bcr-Abl. We've also gone on to show that in Crackle null animals, Bcr-Abl does not transform those cells quite as well. So in fact, it's required substrate for Bcr-Abl transformation. But we use the fact that it's so heavily tyrosine phosphorylated in CML patients to turn this into an assay. So in this assay, we're looking at Crackle phosphorylation. And like most phosphoproteins, Crackle migrates as a doublet. So the phosphorylated form migrates here, slightly slower because it's phosphorylated, more heavily charged, and has a higher molecular weight, so it migrates a little bit more slowly. When Crackle is unphosphorylated, because of less charge, lower molecular weight, it migrates a little bit faster right there. So in our clinical trials, we assessed the status of Crackle phosphorylation either prior to therapy, meaning here where it's mostly tyrosine phosphorylated, or 1 day into therapy, and we can see that even 1 day into therapy, most of the Crackle is unphosphorylated. Now, we can then use this assay to see, is Crackle phosphorylated when a patient relapses? And this is some work from Charles Sawyer's laboratory showing a typical patient who responds and then relapses. So at diagnosis, this is in fact a blast crisis patient. Most of the Crackle migrates as this higher form, the phosphorylated form. 5 days into therapy, most of the Crackle is now unphosphorylated. This patient, in fact, had a very transient response, and at day 22, most of the Crackle is again tyrosine phosphorylated. And this has been the typical pattern of patients who respond and then relapse. Crackle is unphosphorylated when patients respond, and then rephosphorylated, consistent with reactivation of the Bcr-Abl tyrosine kinase at relapse. So that suggests that it's elements in this category. The Bcr-Abl kinase is no longer inhibited at relapse, so it must mean amplification, mutation, or some of these other categories. Now, as it turns out, this category, Bcr-Abl kinase mutations, the target of imatinib is mutated, turns out to be the major cause of relapse in patients who respond and then relapse. So this slide, although it's a little bit complicated, shows you the Abl kinase domain from the N-terminal of the kinase domain to the C-terminus of the kinase domain with the residues that have been mutated. And what you can see, there's a cluster of mutations here in what's called the P-loop or ATP binding loop. And this just gives you an idea of their frequency at which they're identified. There are other mutations scattered throughout the kinase domain. There's one very common one here that you'll hear a fair bit about. This is called threonine 315I. And then others here down in the catalytic and activation loop. So now the first question you'd ask is, okay, so we see kinase domain mutation. Are they really resistant to imatinib? So what we've done is we've taken all of these different mutations that have been observed in patients and expressed them in cells and looked at their sensitivity to imatinib. So here, first of all, just to orient you, this is imatinib. And what you can see is imatinib with unmutated or wild-type Bcr-Abl, very sensitive to cell killing, IC50, meaning the concentration that inhibits 50% of the cell proliferation of about 250 nanomolar. By 1 micromolar, all these cells are dead. But now you can see all these mutations in the curves have shifted to the right. And you can see that there's variable sensitivity. There's this mutation here. This is M351T, which is only slightly insensitive. And it's suggested that if you increase the dose of imatinib, you may see responses if you had that mutation. Here are all the P-loop mutations, and you can see they're relatively insensitive. And then up here is this T315I, which is completely insensitive. So clearly these mutations are resistant to imatinib, and they are driving the resistance that's observed in patients. Now we need to know is, okay, how does this resistance, what does this resistance look like? And the reality is we've been aided by the crystal structure of imatinib. This was done by John Currien's lab at UC Berkeley. And here what you can see in green is the imatinib structure. So there's imatinib, and in orange are amino acids that contact. And here is one in particular, 315. Threonine 315, you can see, forms a hydrogen bond with imatinib. So you can imagine if you mutated any of these direct contact sites, you would have imatinib resistance. And let's look a little bit more carefully at how this threonine 315 to isoleucine substitution works. So here is threonine with imatinib. So here's imatinib, and here's threonine at 315. And you can see it stacks up, makes this hydrogen bond. Now imagine that we mutate this to a bulkier isoleucine residue. And you can see that there's a steric clash. It's too bulky, it goes right into the binding pocket, and imatinib can't bind. What about other residues? And it turns out the mechanism that some of the other residues are a little bit more complicated. So what I want to talk about next are the P-loop mutants. And here I've just shown you a crystal structure of the P-loop. And here you can see the P-loop with an ATP analog. And notice how vertical these two structures stack. So the P-loop, the ATP analog, and they line up just perfectly, and they're vertical. Now let's look at what happens when imatinib binds at this P-loop. So remember, here's our P-loop here. This is what it's supposed to look like. When imatinib binds, it kind of looks like a chair. And imatinib here, which is here, binds at this P-loop and causes this massive conformational change. And so the reality is that this P-loop has a lot of flexibility and accommodates imatinib binding. But look at the residues right at the entrance, 253, 255. If those two residues are mutated, this P-loop presumably doesn't have the flexibility to adopt the conformation to allow imatinib binding, and therefore that's why imatinib resistance occurs with some of these P-loop mutants. Now, the last set of mutants require a slightly more complicated explanation, and I have an animation here that will show you the conformation. So if you look at the activation loop, it has two conformations, closed, open. We'll do that again. Closed, imatinib binds now, and open. So you can see that this activation loop is quite flexible. It can go back and forth from open to closed. And as we'll see, this has a huge impact on the ability of imatinib to bind. So I'm going to go to a static set of diagrams. So here's imatinib. And remember, here's our activation loop. Open and close, just like you saw in that animation. Now here's imatinib, and you can see what imatinib does is it hangs down over this activation loop and actually locks it in this off conformation. Now, if you could imagine if a mutation favored this open conformation, or the activation loop is here in this open active conformation, there's this part of imatinib here that would clash and couldn't bind. And that's presumably why mutations in the activation loop or other mutations that favor this open or active conformation cause imatinib resistance. So now we understand imatinib resistance. We understand mutations. We understand the structural basis of these mutations. So now it's time to do something about it. And the reality is, looking at these crystal structures, there are two things we can do about it. First of all, we can look at the structure of imatinib, and we can actually modify the imatinib structure and make it bind even more tightly than it does. Or we could think about a completely different structural class. Maybe a class that's a little bit smaller, maybe that doesn't care whether this activation loop is closed or open. And maybe that kind of a compound might inhibit some of the common mutations. Now, as it turns out, two major drug companies have taken both of these approaches. Modify imatinib, come up with a distinct structural class. So one of the companies, Novartis, which owns imatinib, modified the imatinib structure to allow tighter binding, and the drug they developed is called nilotinib, or Tasigna, or historically AMN 107. The second company that got into this is Bristol Myers Squibb, which develops Dasatinib, or Sprycel. Historically it was called BMS 354825. This comes from a structurally distinct category of inhibitors that also inhibit the Src tyrosine kinases. As it turns out, there are fewer structural constraints to binding, but because of that, it inhibits more kinases than imatinib or even nilotinib. So let me take you through some of the data for these compounds. Or actually, they turn out both to be more potent Abl inhibitors than imatinib. So here's nilotinib as compared to imatinib. And you can see there's the core structure shared between imatinib and nilotinib. They share this part of the molecule, but sort of this back half is structurally distinct, whereas the front half is identical. So modification of the imatinib structure allows tighter binding. So now we can look at how does this compound do against cells that express the imatinib-resistant mutations. So here in this dotted line, this is imatinib against unmutated Bcr-Abl. And you can see nilotinib against unmutated is way over here to the left with nanomolar potency. And virtually all the mutants fall on this line or below, with one exception, T315I. It still makes that direct contact, so it's still not inhibited by nilotinib. What about dasatinib? As I mentioned, it's a structurally distinct category, and this just shows you the structure of dasatinib. And here is the cell proliferation data. As you can see, very low nanomolar potency against all the imatinib-resistant mutations. Again, one exception, T315I. And again, because it's at the ATP binding loop and because it also makes a direct contact, you can imagine why T315I would also be resistant to Dasatinib. Both of these drugs have gone into phase 1 and phase 2 clinical trials in patients with imatinib resistance, and both have achieved very high levels of response in patients with imatinib resistance. So here, return of blood counts to normal in imatinib resistant or intolerant patients between 84 to 100%. Very much like imatinib achieved in chronic phase patients, where we saw close to 90 to 98% of patients achieving return of their blood counts to normal. And as far as loss of the Philadelphia chromosome or decrease the number of metaphases with the Philadelphia chromosome, again, between 50 to 80% of patients with imatinib resistance treated with Dasatinib. Very similar results have also been observed with nilotinib, and in fact, Dasatinib is already FDA approved for patients with imatinib resistance. So to summarize the Dasatinib and nilotinib clinical trials, we've seen significant activity in patients with imatinib resistance. Activity is observed in all imatinib-resistant mutations except T315I, and much like the imatinib clinical trials where relapse were common in advanced phase patients, again with Dasatinib and nilotinib, the best responses are observed in the chronic phase patients. Good responses are also observed in advanced phase, but relapses are quite common. T315I turns out to be the major cause of resistance or relapse in both of these, with both of these agents. However, there are some other causes. So to summarize imatinib resistance, relapses are mostly due to Abl-kinase domain mutations. There are now 2 novel Abl inhibitors and several others in earlier clinical development that have significant activity against most of the imatinib-resistant mutations. The one exception to that is T315I remains insensitive to all current inhibitors, but fortunately for patients, about the 10% or so of patients or less that harbor that mutation, there are already numerous preclinical compounds, and it's likely sometime in 2007 that one or more of these compounds will enter clinical trials. I'd now like to talk a little bit about some other diseases that are targeted by imatinib. 2 examples are gastrointestinal stromal tumors, which are driven by KIT mutations, and hypereosinophilic syndrome, which is driven by PDGF receptor rearrangements. And in fact, there are a few other rare diseases that are targeted by imatinib that I won't cover. I want to start, however, with imatinib in gastrointestinal stromal tumor. Gastrointestinal stromal tumors are intestinal sarcomas. They actually used to be called intestinal leiomyosarcomas. As it turned out, when patients with leiomyosarcomas were lumped into clinical trials, the patients with uterine leiomyosarcomas, which are the common ones, responded very well to chemotherapy, but patients with these intestinal leiomyosarcomas didn't. As it turned out, intestinal leiomyosarcomas are KIT positive, whereas uterine leiomyosarcomas aren't. So it was very clear that these were clinically and pathologically distinct entities, so they were reclassified as gastrointestinal stromal tumors. The annual incidence of gastrointestinal stromal tumors is estimated at about 5,000 cases per year. As I've mentioned, the response to chemotherapy with these gastrointestinal stromal tumors with very high-dose chemotherapy or multi-agent chemotherapy is less than 5%. In fact, maybe closer to 1 in 200. And historically, if you saw a response to chemotherapy with a gastrointestinal stromal tumor, it was almost worthy of a case report. Now, in 1998, a Japanese group reported that activating KIT mutations were present in the majority of patients with GIST. Now, I remember that paper was published January 1998. I was very excited about this because we knew from our preclinical profiling that imatinib inhibited KIT. And it seemed to me that this would be a very useful tumor in which also to test imatinib besides chronic myeloid leukemia. So I called up the drug company and told them that what I thought was very good news. The first question they asked me was, well, how many cases of gastrointestinal stromal tumor are there? And in those days, it was probably close to maybe 250 to 1,000 at the most. And no big surprise, there was dead silence on the other side of the telephone. I just identified another from 5,000 to maybe 6,000 at best patients, and it still wasn't going to make it over the hurdle of a profitable drug. But fortunately, a number of investigators, including George Dimitri and myself, were able to convince the drug company to expand their clinical trials to gastrointestinal stromal tumor. And once again, rapid, dramatic responses were seen. So a disease that historically had a response rate of less than 5%, with imatinib 54%. And another 28% of patients had stable disease, and these patients also derived a survival benefit. So completely reversing the prognosis for patients with this disease with a simple oral therapeutic that they had to take once a day. Again, responses rapid and dramatic. This is the pre-therapy PET scan. You can see massive uptake in this liver tumor pre-therapy. 1 month into therapy, the PET scan is completely cold. That mass you see over here, no longer visible on this PET scan. The only uptake you see is in heart, kidneys, and bladder, which is just physiologic uptake. Again, rapid, dramatic responses to a targeted agent. Another disease where activity has been seen is a disease called Idiopathic Hypereosinophilic syndrome. This disease is characterized by prolonged eosinophilia, defined as greater than 1,500 eosinophils per microliter in the blood. You also have to exclude other etiologies of an elevated eosinophil count, like parasitic diseases and allergies and a variety of other diseases that can cause an elevated eosinophil count. Plus, you have to see some evidence of end organ damage. So, for example, heart damage, lung damage, brain, or infiltration of the skin. And some of these patients with the infiltration of the skin and other organs can be quite debilitated. Now, as it turned out, there are some anecdotal reports of incredible, dramatic responses to actually quite low doses, 100 milligrams per day of imatinib. Now, the reality is that imatinib had been tried empirically. The reality is the first investigator who tried imatinib in a patient with hypereosinophilic syndrome did it because historically interferon had had some activity, and because historically interferon had some activity in chronic myeloid leukemia, this investigator thought that he'd also try imatinib, and it worked. But nobody knew what the cause of hypereosinophilic was. But it suggested that because we knew how imatinib worked, it targets Abl, KIT, PDGF receptor, that hypereosinophilic syndrome had to be caused by a kinase that was inhibited by imatinib. So from some work with Jan Kool's and Gary Gilliland's lab, they showed that on chromosome 4, where the PDGF receptor resides, there's an intrachromosomal deletion that truncates the PDGF receptor and fuses it to this gene of unknown function called FIP1L1. And what you end up with is this fusion gene that truncates and activates the PDGF receptor. And we know that the PDGF receptor is a good target for imatinib, and by shutting down the PDGF receptor that drives the growth of cells in hypereosinophilic syndrome, rapid, dramatic responses were seen. And in fact, the PDGF receptor in this fusion protein is a little bit more sensitive to imatinib than Bcr-Abl, which explains why slightly lower dose therapy actually works in patients with this disorder. So what this example tells us is that an understanding of the molecular targets of a drug can lead to insights into molecular pathogenesis. And this example I just gave you is actually a reverse of how imatinib was developed. So with imatinib, we understood the cause of a disease, developed a drug, and took it into clinical trials. Here we had an empiric clinical trial that led us to the insights into what causes the disease. And what that also tells you is that if you have a responding patient with a rapid, dramatic response, that patient's trying to tell you something. And what you need to do is you need to study that patient so you can understand why the response occurs, so you can extend that discovery to many, many more patients. So with this part of my talk, I hope I've given you some insights into why resistance occurs to imatinib, but also some of the other diseases where imatinib has worked, and how we can extend the paradigm by careful studies of responding patients. And extending this paradigm and examining responding patients will be the topic of the third part of my talk. Thank you.